do a little bit of background. Um, Introduce myself. I'm Pedro Sanchez. I'm a pediatrician and geneticist. I've been involved in FaceBase basically since its inception, and working with Young Chai and many of the collaborators here just to be the clinical counterpart to a lot of the basic science that's happened. I think we've been doing a lot of collaborations with different phases. I've been part of phase 1 and 2, and hopefully this will highlight some of the work that we've done in phase 3. As a clinician, I'm one of the co-chairs for the Craniofacial Gene Curation Expert Panel, and also sponsored through the NPC and NIH. So, also in the intro, I'm in a new transition. In 2 months, I'll be transitioning to Philadelphia in a new position. So I went from USC to Cedars-Sinai. I've been there for 8 years, and then I'm jumping ship, going back to Philadelphia to have this basically a translational clinician and physician scientist. One of the reasons why I collaborate a lot and I'm on the external advisory board here is, in my sense, does it pass the Pedro test? Like, is this data accessible? Can someone who's outside the loop sort of contribute and submit samples, submit cases, and work with the data through all the infrastructure here? Obviously, there's a lot of talented people working with funded studies. And so can someone who's a clinician or physician scientist sort of work with the data as well? And I think this will be a great highlight of what we can. Almost there. So I think we've got a nice setup. So that's basically the background a little bit. I'm going to do the ClinGen part, and then Jifan will do some of the interactive examples through their Atlas. And then we'll talk together about future directions and hopefully have an additional discussion. So ClinGen was founded in 2013, and there's 59 expert panels. I think there's an ear expert panel, as you heard the previous talk about gene therapy and how do they get through all these genes and prioritize them. So every expert panel has their standard operating procedures of how they review genes and assess variants, sort of prioritize them. We get through maybe 2 or 3 genes per month. And so there's so many genes to go through, but we're standardizing the process. And ultimately, we go through the low-hanging fruit, easier, well-defined syndromes so that we have a good discussion. We do some education and teaching. And at the end of the day, once we have these procedures in place, hopefully we can automate some of the assessments of genes and the profiles. So my job right now is the gene disease variant curation. One gene can have many syndromes associated with it, some with a lot of evidence, and some that evidence is sort of refuted. We look at the papers, we ask and invite experts that have published this to join the conversation. So we have our consistent group, and then we have people who sort of join, on an as-needed basis. Ultimately, this is all really to get some of this work into the clinic. So as you can see, from the initiation of the whole genome sequencing projects that people have started in 2010 and onward, people have moved away from gene panels to really sequence everything and assess them on a clinical basis. So we bring together patient data, research data, some clinical profiles. A lot of laboratorians joined the call, and clearly a lot of basic science is presented. So in our working group domain, my pitch today is if you have someone who's undergraduate, or even I've had 2 high school students join us, learn to do gene curation, contribute somehow. If they're ready, they could present to the group for 1 gene and feel accomplished, feel integrated into the team. We also accept students during the summer. We train them and we integrate them. So we've gone through a couple rounds of gene curation. We start off with just saying, is there enough evidence to include it as a gene that we want to have a discussion about? If there's only been 1 paper with 3 patients, clearly there's not enough discussion, but some of them have gone through many years of discussion and integration already in the clinical workflow. So we move that into, we plan maybe 1 or 2 years in advance with 20 genes. We move that into the data gathering phase where we have a biocurator and their team work through and clean the the information and sort it in a standard process. Then when they're ready, they present it in a pre-curation meeting, and then we move it on to the full GCEP or the gene curation expert panel, where we discuss it. We give each component a certain number of points, and if it passes a threshold, it will get published on the ClinGen website. So in our round one, we had a lot of craniosynostosis experts, and really everybody has their preferred pathway of how to assess a patient. And so we might use one gene panel for one patient if it's clearly a recognizable syndrome, or use a broader test if it was just a non-syndromic form of synostosis. And really, it wasn't clear exactly how each lab picked their genes. And so I also was part of a lab that made our own curation, and we wanted more on that panel. So we might pick, 84 genes at the bottom, and just, at a certain point, the evidence from some of those genes are less and less. But at least when we picked our first round of genes, we picked the core genes that are common amongst all profiles. It was a very craniosynostosis heavy expert panel. So we wanted to integrate other providers, people who did cleft palate research or craniofacial microsomia or mandibular facial dysostosis like Treacher-Collins syndrome. And so we curated some of the more common genes and had a broader discussion. And later on, you'll hear how we've integrated some of the discussion amongst our dental experts. So when we present it, we get the clinical evidence, basically case level presentation, phenotypes, segregation. Is it a recognizable syndrome or is it more of a non-syndromic? We have a nice lumping together discussion or splitting apart discussion. If there's any clear genotype to phenotype correlation evidence to say, well, this is a recessive form of the same gene. It's more severe, so it's more defined. We have a nice discussion, but we also have experimental evidence that we need to integrate. Gene level experimental data. Really, we try to integrate both because both have more strength in determining whether or not this gene is classified as definitively related to human disease, or there's limited evidence. Maybe there's only GWAS data, or maybe there's one publication and it's been refuted evidence or disputed. We've also characterized that. So each GCEP for each different group has their approach. And I think for the craniofacial category, you'll see that other people's approach have to be modified specifically to the craniofacial GCEP. And so this is what we've published already online. Some of them are clearly gene disease relationships with a definitive correlation, and some gene disease relationships have been disputed. And so we're getting through more and more genes. We just published last week the first craniosynostosis round 1 gene list, and most of them did get moderate or definitive classifications, but some of them were more limited. And so how do we move them across a little bit more? Either doing individual level data or trying to get access to craniofacial specific gene expression. And so that can play a big role, but up until recently, we didn't have clear expertise to getting access as a clinician and as mentoring undergraduates to really curate these genes on an regional or craniofacial specific review. So I have 3 questions that I asked Jifan to help me sort of compare with FaceBase data and the ClinGen data. This is one of the families that really changed my practice completely. 2 sisters with cleft lip and palate, isolated and non-syndromic. I really didn't know what was the underlying cause until the mom came up to me and said, I was diagnosed with stage 3 stomach cancer. It was already metastatic, and really she wanted to know if that was genetic at all. And there is a gene, ACAN one, that causes diffuse gastric cancer. And so I looked up gastric cancer and cleft, and right away this came right up to the top of the list. We sequenced her and her 2 kids. Unfortunately, she passed away, and both of her kids have the same variant. Fortunately, you can do surgery and basically do a total gastrectomy to reduce the risk of stomach cancer. But now I have a mom who passed away, and we could have gotten that diagnosis early. So now we're sequencing all patients very broadly. When we see kids with cleft lip and palate, I've had 2 moms pass away of this, and it's really it's not just moms, but it's a dominant condition. But the real question is this gene a cleft palate gene? There is some evidence, but when we go to gene expression, I just want to know, is this gene in the craniofacial complex? Like I go to GTEx, I go to all these repositories of gene expression, and doesn't have the resolution that we really need. So the second question, and Jifan will answer that for us. But when we curate mandibulofacial dysostosis, and clinically we know that there's this gene-gene interaction. We know that there's a gene-environment interaction. But can we, look at a severity scale using 3D images? We heard some presentations earlier yesterday. And yes, there are heat maps that you could do just from 2D pictures. Can we get some of that interaction with the genetic data and the morphometric data to look at just, are these genes expressed in the same pattern or not? And so again, publicly facing data on gene expression, it just doesn't go to the resolution that we need clinically. And finally, I like using the use cases. Can we get some of the data as a clinical side, put it into the FaceBase system and have it be usable for other researchers? And so this is a patient on the left side, your right side of the screen with a U-shaped cleft palate. She has a large head, short stature. The dad is large head, short stature. They have advanced bone age. And clinically, this is non-syndromic short stature, but really she has an ACAN mutation, which causes short stature, and it's a chondrogenic gene. But my patient has a cleft palate. Is this the cause of a cleft palate or not? Is this gene at all expressed in the palate? I poll all the people that I know clinically, and they're like, well, I have one case. Maybe the palate looks a little different, but didn't need surgery. She clearly needed surgery. So I put her through, this family, through the FaceBase system. We enrolled them in the rapid genome sequencing. So all their data is in the human side of the FaceBase database. We got 3D MD imaging. This 3D morphometric data is accessible to you. We could definitely ask the question, is her jaw smaller? Is that why her palate might have been more of a Robin sequence secondary effect of cleft palate, not necessarily a primary effect if the gene isn't even expressed in the palate? But my question is this a palate gene? And I have a little bit of evidence to say, in 1978, there was a mouse model of this gene that had a cleft palate. There is some expression, but again, not to the resolution we need. And there is a miniature horse that has a palatal issue. So I'm finding evidence, but again, I don't have expression data and I'm not a basic scientist. So I want to use FaceBase for craniofacial research. And again, GTEx has some expression patterns, but for our gene curation, it's not craniofacial. So it'd be nice to really have our biocurators access FaceBase and really just know, is gene X expressed in the craniofacial complex? Yes or no? And anything above and beyond that is great, but it's just can I see it or not? Because I'm a visual type of person. Can it be quantifiable? Is it not even there? Is it low expression, or is it all over the place? And then can it be co-localized? This is the pattern. And so Jifan is going to do the highlight of the talk.
Thanks, Pedro. Good morning. My name is Jifan. I'm a research scientist from Dr. Yang Chai's lab at USC, and also a member of the FaceBase team working on this Gene Expression Atlas. So I'm very excited to share that we are introducing the beta release of the FaceBase Spatial Transcriptomics Gene Expression Atlas, which will provide anatomical structure-specific gene expression information for disease and development-related genes. So we are able to host this atlas on FaceBase because FaceBase has this can support diverse data types across FaceBase can support diverse data types across species, and it has the right infrastructure to support various imaging formats, including those needed for the spatial gene expression atlas. So we take advantage of the powerful FaceBase platform and the recent advances in spatial genomics technology, and build a hub for the spatial genomic data sets related to the craniofacial region. So we curated and consolidated spatial genomic data sets related to the craniofacial region from both FaceBase and other public data repositories. We then process this data to generate this gene expression atlas so the users can conveniently browse their genes of interest. And if they find anything interesting, they can further use the direct link we provided to access the raw data to perform more comprehensive bioinformatic analysis. Currently, most of these steps are done manually by our FaceBase team. And so we are working towards incorporating AI into this data curation and processing process to speed up the data curation. So this atlas is still at an early stage as we continue to incorporate the new data sets from FaceBase releases and the new publications. Currently, we have a spatial data sets covering the whole embryonic head, as well as focusing on the palate region. And we have incorporated both imaging-based and sequencing-based data to complement each other. So for the imaging-based silkfish and murfish, they have exceptional spatial resolution as far as single molecule level, but the target genes are restricted to smaller pre-designed panel. So for these two data sets, we have included the full gene panel from the original study. For the sequencing-based Pizza HD and upcoming AeroSeq, they have a lower resolution but cover the whole transcriptome. So for these data sets, we are currently offering selected gene panels related to disease or by request. For example, we have the craniofacial malformation panel that Pedro is working on. And we have also included the tooth-genesis, cleft palate, the gene panel from HBO. And we're open to collaboration and working with you for other gene panels you are interested in. In the meanwhile, the FaceBase team is also working on updating our viewer so we can accommodate more gene channels and eventually can accommodate the gene channel at the whole transcriptome level. So to address Pedro's first question regarding the in vivo expression pattern of ACAN in the palate region, you can just type ACAN in our gene search bar and then click search. This will bring you to the data browser showing all in vivo gene expression data containing the ACAN gene. And you can further browse this data based on either the developmental stage or the tissue, and then click on the thumbnail of the data of interest to access our interactive viewer, which is designed to mimic a microscope-like experience as Rob demonstrated yesterday. So we have to facilitate the exploration of tissue-specific or anatomic-specific gene expression. We have included annotations for key anatomical structures such as the palate, tongue, mandible, and the tooth. And wherever possible, we also included the marker genes such as the epithelial cells showing cyan, the bone cells showing blue, dental cells showing in magenta, and the muscle cells showing green to help explore whether the gene of your interest is expressed in the known cell type of interest or not. So to check ACAN expression in our browser, you can use the channel panel on the left and turn on the ACAN channel, which is showing in yellow here. So you can see that in low magnification, there are some low-level ACAN expression in the palatal shaft away from the bone and the dental cells. And we can even zoom in to examine more details as fine as a single cell level to compare the expression of ACAN with other marker genes. We can also use the gene expression atlas to compare the expression pattern of a group of genes associated with trichocolon syndrome, as Patrick mentioned earlier, to see if they are co-expressed in the same region or not. So we found that the trichocolon syndrome associated gene TCOP1, shown in yellow here, is expressed in the maxillary region, including the dental and the palatal area. And so are other genes such as POR1A, POR1B, 1C, and 1D. And we can overlap these genes using different colors and confirm they are co-expressed in the same region. So since this is a sequencing-based data set, so the resolution is not high enough to draw definitive conclusion whether they co-localize in the same cells or not. However, this provides useful preliminary information for further investigation of this co-expression as well as potential synergistic functions. So with that, I will hand it back to Pedro.
So I think it answers the question that we have. Co-localization, is it expressed at all in the palate? And we could prioritize genes. We've now moved on to round 3, where we have a dental phenotype looking at 560 genes. Really, it'll take years and years to get through them. But can we use this data to prioritize our gene list? Really, is it expressed at all in the tooth? Is it tooth specific or is it tooth plus? And then can we use this data to inform the clinical side to say, you know what, this tooth has a palatal expression. Can you just go back and look at the uvula and the hard palate and see if there's any kind of phenotype there? And I think we'll see that the answer will be yes.
So we will use the tooth agenesis gene panel as an example to show how we can use FaceBase Gene Expression Atlas to help prioritize genes for the ClinGen curation. So in collaboration with ClinGen, we have designed a workflow to systematically screening these genes step by step. So we first ask whether these genes are expressed in the tooth region or not. So we have found that genes such like ATCL6B are showing color magenta here, are not expressed in the dental region, as we don't see any of this magenta signal here. So for genes like this, we will exclude them from the list because the lack of activity indicates the lack of function. For the genes expressed in the tooth region, we then assess whether the expression is specific to that region or broader across tissue. And we were able to identify genes like TNF, also showing pink here, are expressed not only in the tooth region, but also in the palate and in the tongue. So this broader expression suggests that these genes may have function beyond the tooth. And then Pedro can revisit clinical record to determine whether other regions also affected in his patients as well. Then, since the tooth consists of the epithelial and the mesenchymal tissue, so for the tooth-specific genes, we further ask more specifically, are they expressed in the epithelial, mesenchymal, or both? So, for example, we have identified genes like MS1, also showing magenta here, that are expressed in the but not in the epithelium, suggesting they regulate tooth development specifically through the mesenchymal sulfate, not through the epithelium. So currently we are doing this screening almost manually, but with this workflow already in place, we can see the possibility to integrate AI to automate this gene expression data integration to speed up gene curation process for ClinGen and Pedro. So with that, I will hand the final thoughts.
So I think watching Jifan do this really took a lot of work to get the anatomy defined, to really work through where the data is and how to turn it on, and even picking different colors for X number of genes. But once we have our workflow, can we work with Rob and that chatbot to say, you know what, use example number 1 for the palate, example number 2 for tooth, And we would start training the set and saying, okay, we like this, do this over again. And I think as we do this more and more, it becomes a very useful tool for us, mainly for the craniofacial GCEP. But if we present this to the greater ClinGen group with all the other ear profile and gene expression and kidney profile and gene, brain, like this data will be very useful once you visualize it in the right setting. So, we gave a little example of just what would you say to a chatbot? Show me a coronal image of these 5 genes using different colors, in the rainbow. And really in this region, I'm specifically to that region. Once we define where on these images, where do we want to focus on, then it'll be something that even someone like me, a clinician, can start using and find, how to integrate that in our curation. So wanted to open it up to questions. We had a really great working group yesterday, did a live demo. And for the people who were online and everybody in this room, I hope you're just as excited as we are.
Yes, Lou.
Yes, Pedro, you already answered my question about other ClinGen working groups focusing on different health conditions. So asking my second question now, that is, with the experience so far with, the spatial genomics data and the FaceBase experience to really view and sort of doing the in silico experiment of looking at the co-localization of different genes at a given time point. What's your sense of the sensitivity of the combination of the wet lab result and the FaceBase visualization for genes, for a disease condition that is polygenic and likely involves the expression of multiple genes at very low level. So if you are dealing with genes expressed with low abundance, but together, given the polygenic nature, and still cause a disease phenotype, do you have a sense of how far we can go at this point? 10 genes expressed in as far as, as few as just one cell at a given time?
We talked about it a little bit last night. And, for us as humans, we only see a certain spectrum of the color of the visual field. And so, but a computer has the even broader scale and really could differentiate, like the UV spectrum, or it doesn't have to be visual. And so I think really just having the data is key. When I looked at the ACAN example, I actually went to Axel initially and I'm like, is there an enhancer that the child inherited from mom? That's different. That data is in FaceBase. I mean, we can mine it for these questions one day. Right now, Jifan has wonderful control data. Did a little bit of environmental exposure and then did gene expression and then uploaded that. It really is just, first is, can we build a foundation that's strong and then populate it with all these data sets?
Yeah, so if we could get more and more data, for selected disease conditions, that would be great.
We have the platform ready.
Right.
I believe with the advance of spatial genomics and more and more group are gonna use this
Exactly.
We will have more data coming.
Yeah. I'm saying this also to Susan and my director remotely.
And the same goes for expression changes over time too.
Exactly.
So you just need the data there, and then even if it's not visualized in color, computer can find the changes.
Right. And if it's not expressed, if a gene is not expressed in teeth at time point A, what about time point B?
We just need the data.
So that leads me to think about the disease onset.
Yeah, and that that data is there. Jifan has some earlier data and images. The real question is, can you explain it to the group of the anatomy that you're really looking at? And so it's great to tag team with someone who knows the mouse anatomy to immediately point out those structures and then the stages. So there, that to be continued file
Yeah.
is more like that 3D spatial
Yeah.
visualization of where the palate starts, the hard, the soft. Like
Yeah.
there's a lot of work in progress, but my main goal was that the FAIR principles are, is it findable? Like,
Yeah.
is it accessible? Like, and it shows that it's really a tool already that I'm definitely gonna present to the ClinGen.
Awesome.
Thank you.
Thank you so much.